overall, of course, it's a rare entity, as all pediatric tumors. So we basically, all pediatric tumors together are about 1% of all cancers. But amongst the pediatric tumors, the brain tumors nowadays are the leading cause of mortality or patient death, and this is why we are so much interested in improving their situation. Basically, after the primary therapy that we give to all patients, usually in nationwide or European protocols, we nowadays can cure about 75% of patients, and whoever relapses, however, usually has a very poor survival. So we decided that we have to do something for this group of patients, independent of the tumor entity that they are suffering from. And we try to foster a rebiopsy at the time of relapse to really then do genomics analysis on the rebiopsy in the relapse to be sure that we are actually facing the biology that is present at that time, rather than taking the biopsy from the primary tumor. And then we try to infer targeted therapies from this molecular workup that we are doing for the relapse situation, because we think, or we hope, that this will be better than just giving a standard oral chemotherapy. As you can imagine, that these children are treated in more than 50 different places in Germany, and about 12 different study centers are involved in that. So it was kind of challenging to get all of them together. But I think that we are all unified by the wish of better helping these patients. And we set up an infrastructure now where in all of these 50 places, tumor tissue can be frozen down and submitted to a central laboratory, which is in Heidelberg. And we try to turn around these samples within 4 weeks between the biopsy and delivering the data to the treating physician. And yeah, we try to provide rational drug targets that then, at the moment, it's at the discretion of the clinician, the local clinician, to take them further for an individual therapy. And in the future, once this concept is better established and all the infrastructure is working really on a daily basis, then we want to really run a clinical trial where we compare the principle of individualized therapy versus just giving a backbone chemotherapy, as we do now. So far we have, and this is kind of encouraging, we have done about 30 pilot patients, because the registry study as such is not open at the moment. We just started to establish the infrastructure, and amongst these 30 cases, we have identified or been able to identify drug targets in more than half of the cases, which we think is quite good. And now it will, of course, be interesting on getting the feedback from the clinicians. We already know that we had some very favorable responses, but we cannot really talk about frequencies and durable responses yet, because it just started last December. We even have I think more limitations to get access to drugs, of course, because usually you have to have completed an adult phase 1 study before you can start a pediatric phase 1, and usually there is not a lot of interest with the pharma companies to do that at all, because this will never be a big market. But on the other hand, we try to find different ways on getting access to drugs, and one of, I think, the most convincing arguments from a pediatric oncology perspective is that usually the genomes of children's tumors are relatively simple, so you're very close to the actual driver mutation, and this is also an attractive option for a pharma company to really give a drug where the target is actually present, and there's not 100 other potential targets which probably could mask the effect of this drug. So we hope that with this argument we will convince more and more companies to be interested in that. Yeah, as I said, the genomes are simpler, so it will be much more much easier to interpret the results of giving a targeted drug, even a monotherapy, and probably the mechanisms of resistance. Yeah, because it's just less noisy and less what we call passenger mutations. Of course, inferring kind of targets and drugs from the genome sequencing data is just it's a very far distance to bridge, right? And in principle, you should have a kind of functional genomics or approach for every patient in between, where you would do some kind of in vitro or in vivo drug testing to really show that this is working. And so this is something where we are currently trying to really fill the gap, and for the most interesting and common targets, we also try to really do some more in vitro and in vivo work prior to giving it to large cohorts of patients. Well, there's one thing that I also mentioned in my talk was that especially in these kind of relatively simple pediatric tumors, we often have cases also where we haven't identified a clear driver mutation at this stage even, and we think that and we have good evidence now that if you integrate epigenomics information rather than only looking at the pure mutations in genes, that we will be able to really fill these gaps of cases where we don't find an obvious driver. And this is something that will probably also be interesting for adult oncology, because this is something that is probably not a mechanism that is restricted to pediatric tumors.